Okay, so we've talked a lot about the resting potential, right? Which is how a neuron, expends a lot of energy to sort of maintain differences across the membrane in terms of both charge and in terms of concentration of the ions that carry the charges, right? And that sort of sets up the neuron so that, should it encounter a stimulus, right? Like an excitatory stimulus. That could be, for example, in the brain, right? At a synapse, should one neuron release what we call an excitatory neurotransmitter, right? A neurotransmitter like glutamate, for example, that's the most common excitatory neurotransmitter in our nervous system. Should glutamate be released from the axon terminal of one neuron, it might bind then to a protein, what's called a receptor protein here, postsynaptically on the next cell. And remember, when one chemical binds to another, there's a change in conformation or shape. Well, it could transiently open a channel, right, that would allow for ions to pass through, let's say sodium ions, and then sodium will, come in, driven by the concentration gradient and electrical forces, right? And it will, lead to depolarization. There'll be, more positive ions coming in, to the neuron that was at minus 65, before the glutamate neurotransmitter bound here. But now suddenly there's more sodium, moving from the outside to the in, driven by electrical and concentration gradient forces, and that's leading to depolarization. And it could also be, for example, we're going to learn in perception, right, that if you take your finger and you touch right here, actually here's an example of one of these cells. There's a cell here that's it's got this little ending here, it's like this little corpuscular ending, and it'll push ions and, push the membrane of these specialized afferent cells. And there are proteins that are embedded in their membrane that in response to the actual pressure will twist open holes or channels that will let sodium enter the cell, and that'll lead to depolarization of this thing called a Meissner's corpuscle that we'll talk about later in the class. But so that'll also lead to sodium, crossing in, and depolarizing, right? You're bringing more positive ions from the outside, into the inside, where it was at minus 65 millivolts at the resting potential. And so you're going to be making it less negative inside versus the outside. You're going to be depolarizing, reducing that charge difference. And we're going to see depolarization, whether it's because of an excitatory neurotransmitter or whether it's because of, some kind of, specialized afferent mechanism that, couples detection of whatever stimulus, whether it's, olfactory or, taste or, whatever, pressure, touch. To depolarization. That's going to be really an important component typically, for many of our sensory systems. But that's going to be most likely to lead to what we call action between the potentials, an action potential, and the initiation of a current, right, that can run down the length of the axon to its end. So the action potential, what is that? It's how electrical messages travel within neurons. And, basically it's a rapid change in potential difference between those potentials, right, charge difference, across the neuron membrane that's caused by the movement of ions. So ions move across membranes, right, not directly through the phospholipid bilayer, because that's impermeable, right? It's got that fatty, those fatty tails that make it like impermeable to the passage of sodium and potassium, but through specialized protein machines that are embedded in the phospholipid bilayer. And at the resting potential, we talked about how important the non-gated channels are, right? The ones that are always open, the leakage channels for sodium, the leakage channels for potassium. But it turns out that depolarizing that membrane, right, to a very specific value, to around minus 55 millivolts. Actually, that's a depolarized value, right? From minus 65, we call minus 55, that depolarized value, we call that a threshold potential, because there's a lot of other machines that are embedded, a lot of other protein channels that are embedded in the membrane of a neuron, but these are what we call gated channels. So it turns out at minus 65, at that particular voltage, that particular charge difference, that particular level of polarization at rest, at the resting potential, they're all gated and they're closed. But should you depolarize, right, the membrane, again, because you pressed here, you opened, channels, sodium channels, and you've allowed sodium to come in and depolarize, or because, you've released, neurotransglutamate, and it's opened sodium channels over here and allowed for depolarization. If you depolarize to minus 55 to threshold, these voltage-gated channels, right, depolarization-gated channels for sodium will suddenly ungate. And there's many, many, many of these actually packed all around the axon hillock, which is the initiation point, for an action potential. There's lots and lots of dendrites here, right? Lots and lots of, let's say, synaptic connections, right? We're going to see some of these maybe resulting in hyperpolarization, right? They could open, potassium channels, for example. Some of them may be resulting in depolarization. But if you get enough, sodium in here to depolarize at the hillock to the threshold potential of minus 55, well, you're going to open a whole bunch of these voltage-gated sodium channels. They're going to swing open like this and allow for sodium to pass from the outside to the inside. Now, these voltage-gated sodium channels are really interesting because they have a number of distinct, we call conformational states or structural states that they can assume. So this may seem kind of silly, but this seems to help people kind of remember. This helps me remember, what the sequence of events that's occurring here. So let's say something takes a neuron and depolarizes it to minus 55 millivolts from the resting potential of minus 65. So minus 65 is what, remember that the neuron's working really hard to maintain, spending a lot of ATP to maintain, a certain level of charge difference at minus 65. That's the resting potential. It's resting. It's a very energetic rest, but it's staying the same at minus 65. But then something happens. You press, there's excitatory neurotransmitter, an excitatory synapse. Sodium channels are open. Sodium comes in. And you depolarize that membrane now to minus 55, to what we call the threshold potential. Well, minus 65, this is the voltage-gated sodium channel. Pretend I'm on the intracellular side. This is the extracellular side towards you. It's gated, right? This channel, this voltage-gated sodium channel, is gated and closed. But at the threshold, at minus 55, it opens the threshold, right? Suddenly there's a hole that goes, from the outside to the inside, right? And sodium, driven by the concentration gradient and electrical forces, just rushes inside, right? And this thing is voltage-gated, right? It's sensitive to the charge difference, the voltage across the membrane, right? And again, minus 65 closed, minus 55 swings open. So much sodium will come in that it'll suddenly go, continue to depolarize to minus, 45, 35, 25, it goes to 0. It actually overshoots. It reverses polarization. It suddenly becomes positive 20. When it hits a sort of positive value there, this thing again responds and shuts. It shuts, though, in a very different way than it was shut at minus 65 at the resting potential. Now we're going to see that it'll stay closed, until the neuron is repolarized, brought back to the resting potential, when it will go and reset and be ready to go again should you depolarize again to the threshold of minus 55. So the initiation of an action potential, right, the start of current, right, action between the potentials is depolarization, from the resting potential of minus 65 to the threshold potential of minus 55. Because here's 65, at 55, it swings open, right? And then at, so much sodium rushes in that it becomes positive 20 suddenly on the inside relative to the outside, and then this thing closes. And then now we're going to have to repolarize. We have to get back to the resting potential, right? And then at the resting potential, this thing will go and reset and be ready to go again should you depolarize back to threshold of minus 55. So how do you repolarize? Well, when you hit, the threshold of minus 55, when the neuron is depolarized, to this threshold potential, not only do the voltage-gated sodium channels open, but there were also these other voltage-gated channels for potassium. And the voltage-gated potassium channels open, And the potassium will actually, go right from the inside, remember, to the outside. Remember, the concentration gradient force wins at minus 65 at the resting potential, right? Well, at the threshold potential, when you've depolarized that neuron down to minus 55, well, the electrical force is even weaker, right? So it'll go out even more, right? It'll go out. So basically, you're taking a positive ion, positive potassium, and you're moving it from the inside to the outside. That'll help to bring down the voltage, right? The voltage will start to, go back to, minus 65, towards minus 65. The other contributing factor, right, is that the sodium that rushed in, remember, at minus 55, and it's positive 20 here, is not going to stick around here because it's in a high concentration now on the inside, right? And a little bit further down the membrane, it's at the resting potential where it's negatively charged, so opposite charges attract, right? And there's no sodium because there's only leakage channels down there, right? So it'll move, down the membrane and get out of here. So this will help the opening of the potassium channels, the exiting of the potassium, and the exit, the sort of movement on the inside of the membrane of the sodium down, away from this area, will all lead back towards a repolarized value of minus 65, right? It actually overshoots a little bit. It goes to like minus 70 or so, or 75. And then the pump keeps working, and then you work your way back to the resting potential. But that will, reset those voltage-gated sodium channels so that you can initiate another action potential again.